Oh, good. So we're up to active transport. Oh, good. Excellent. All right. We have moved very quickly through these slides because it is revision of last year's stuff, and it's going to continue with active transport. So, what are the situations where active transport might occur? When a molecule is too large, so it can't go across by itself. Yeah, definitely. And? Going against the concentration gradient, yeah. So if we need to push a substance against its concentration gradient, so moving from a low concentration to a high concentration, or if it's particularly large. Because of the nature of it, that it uses energy, it's actually possible that it could be exhausted. With facilitated diffusion occurring through carrier proteins or protein channels, you don't exhaust it, you saturate the proteins. They don't actually tire out, they just get so full of the substance if the concentration gradient is too great that they actually just plateau. You eventually reach a point where the diffusion rate can't actually occur any faster. With active transport, although it's possible to saturate the proteins, it's also possible to exhaust the energy supply. So you can get a diffusion rate that actually plateaus, but you can also get one that starts to go backwards. Or not backwards, but starts to drop. So if you run out of ATP, then the protein pumps or the endocytosis or exocytosis, the change of shape of cell membranes, isn't going to work. We're not going to do that activity today. We'll deal with that later. So we're talking about endocytosis and exocytosis. Very large molecules, so large proteins, large polysaccharides, all those required in extraordinarily large volumes, cannot move via membrane or via proteins. So these require a bulk transport system, and that system is called endocytosis or exocytosis. Endo for going in, and exo for going out. If you remember exit and entry, that's the simplest way to remember it. Endocytosis takes substances into the cell. Within the small intestine, it occurs among other places, but in the small intestine or macrophages. With macrophages, it's called phagocytosis, and it's phagocytosis is particularly directed towards cells and cell products of non-self organisms, so bacteria and other sort of organisms that might exist in the body. Exocytosis is taking substances out of the cell and allows things like secretion of hormones from cells or secretion of digested cellular debris into the extracellular fluid. Particularly with hormones, when we're thinking of those, it's the big one that we refer to regularly are neurotransmitters. They're released via exocytosis from the axon terminus of one nerve to the dendrite of the next nerve. So here we've just got diagram or figure again from your textbook, figure 248, I don't remember the page. This is of endocytosis, so we're taking something in. This is a simplification. One of the things I did want to point out is that, oh yeah.
I just want to ask a question.
Yeah.
Endocytosis uses energy, right?
Yeah. Since it's active, yeah. The energy isn't like with protein pumps where the ATP is utilized directly by the protein pump itself. With endocytosis or exocytosis, the energy is used by contractions of cytoskeleton. So it's actually the fact that the cell membrane itself has to change shape, and that changing shape occurs via contraction of cytoskeletons or cytoskeletal proteins. This is still a selective process. Endocytosis and exocytosis are selective, so it's not just that the cell runs into a substance and ingests it, it actually needs to identify it. So what part of the membrane, which of the components of the membrane is going to control that selectivity? Yeah, the protein receptors, exactly. So protein receptors that are specific for that particular material, that particular substance, they are the ones that are going to drive endocytosis, and on the flip side, exocytosis. With endocytosis, the cell membrane gets very fractionally smaller. With exocytosis, it gets very fractionally larger. Now, the net result is we sort of assume that the rate of endocytosis equals the rate of exocytosis as an average. That's probably not feasible for a lot of different cell types. Like if we consider cells like cells in the small intestine, they would certainly be undergoing far more endocytosis, at least the cells of the microvilli. They'd be undergoing far more endocytosis because they're taking lipids in, or they're taking large amounts of carbohydrates or amino acids or whatever. They're taking those in, and so we would assume in those sorts of situations that endocytosis would be occurring more frequently than exocytosis. Whereas on the flip side, a cell type or an organ that is producing large amounts of hormones, so cells or organs like the pancreas or nerve cells, they're going to be producing large amounts of hormones, which are going to be undergoing lots of exocytosis, and so they are quite likely to actually be increasing in size. So there's this need for a constant balance. Cells need to maintain a certain size, and what's driving that need for keeping that size? What's the selective pressure that's there? If the cell gets too big, what's going to happen to it? Yeah, but it's the other part of that surface area to something ratio that we're concerned with. Volume, yeah. So if it keeps increasing its cell membrane, it's also going to increase its volume, which is going to decrease what? Rate of diffusion, or certainly efficiency of diffusion is going to decrease. So it needs to maintain the size of its cell membrane. So although on the surface we just think, okay, this is just an active transport process, there's also this additional thing of, well, if the membrane is actually changing size, what are the kinds of consequences that that might have for the cell? So it needs to have some kind of way of managing that. I don't remember what that is. Let's have a quick look. Oh, this is useful. Oh, okay. We'll wait on that one for a bit. We'll finish off the next few slides. Yeah, we'll get to the key concepts and then we'll do that. All right, so factors affecting the exchange of materials. So obviously nature of the membrane, this has already been discussed. It's a major determinant of the material exchange, but there are obviously others. So surface area to volume ratio, concentration gradient, nature of the substance to be diffused, all impact the rate of diffusion as well as the likelihood of diffusion occurring at all. So those are three others. These are separate to those characteristics or those features that we talked about where we were looking at things like the environmental factors, so temperature and those sorts of things. These are factors that directly affect that Fick's law. We talked about surface area, concentration gradient, distance to be diffused, those sorts of things. That's what we're talking about here. And each one of those relates to that in some way, membrane permeability as well. So first one, surface area to volume ratio. Surface area to volume ratio is a relationship between the surface area of a cell's membrane with the volume of the cell. Now, we don't need to go into the exact mathematical details, but volume is cubed, surface area is squared. So for each value that the size of the cell membrane increases, the circumference, the diameter, of the cell's membrane increases, the surface area will increase by far less than the volume. It's an exponential difference. So we actually have a far more massive increase in volume for every increase in surface area. This is what we're really talking about. This is the problem. So the larger the surface area to volume ratio, the more surface area relative to volume is available for exchange to occur. If we have, I know this isn't three-dimensional, but if these are our two potential cells, the larger one has far more massive surface area. So it is actually, by volume, able to undergo more diffusion. More substances can cross into that cell. So more molecules can cross into it, yes.
So would it take longer?
Yes. Because there's always a target for these molecules to get to, it will be less efficient. The greater the surface area, the more molecules can actually enter into the cell, but it will be less efficient because the volume is increasing at a far more rapid rate than the surface area is. Does that make sense? Again, it's something that we've covered a few times last year, and we'll talk about it more as we go on. What that ends up meaning, this is just a representation of that. This is again from your textbook, but you could find this exists almost everywhere. What this ends up meaning is that there is a limit on the size of a cell. It's a theoretical limit, it's not a precise limit, because obviously we're going to encounter manipulations, we're going to encounter adaptations that allow for an increase to that. But there is a theoretical limit to the size of a particular cell, dependent on its energy requirements and oxygen requirements and waste removal and all of that sort of thing. So this relationship surface area to volume ratio actually has quite a direct impact on the physical nature of the cell. It also has an impact on the size and shape, and not just the size, but the shape of the cell. Because if a cell changes shape, so a sphere has a particular ability or a particular surface area. If we flatten that out, so the volume remains the same, but the cell changes its shape, then we're going to have a different relationship between surface area to volume ratio. It's still better to be smaller, but by changing shape, we're increasing the surface area while keeping the volume the same. Does that make sense? So a lot of adaptations revolve around that are about trying to minimize the impact of an increased volume are actually directed at changing shape. And we look at that throughout our bodies in our organ systems. We look at the microvilli in our intestines. We look at the alveoli in our lungs, the capillaries in our cardiovascular system. All of these things, we're not affecting the overall volume of air that we can intake or the amount of food that we can digest, the amount of food that we can fit in our stomachs or in our intestines. What we are affecting is the surface area by changing the shape of those structures. When we look at something like concentration gradient, this is just the size of the difference in a molecule's concentration on either side of a membrane. And again, this is very much revision sort of stuff. Typically, the greater the difference, the faster diffusion may occur. What might be an exception to that? If you've got an incredibly massive difference, so a very steep concentration gradient, what might be a situation where diffusion can't occur any faster? Yeah.
So if the molecules are too big?
Yeah. So if there's something to do with the actual molecule to be diffused by itself, or if the membrane is also going to not accept, not allow, so if the membrane is impermeable to the substance, or we might get to the point where it's being diffused by facilitated diffusion by a carrier protein, and all of the carrier proteins on the cell membrane actually become saturated. If a cell experiences that saturation, what's its likely response? Long or medium term? Not instantly, but medium or long term.
To get to its original.
Possibly, kind of. I'll use the cell that I've got up here. I'll use the smaller one because it will mean I'll have to draw less. But so, let's say we've got our carrier protein, and this cell has 4 carrier proteins. So we've got 4 carrier proteins, but there are 20 molecules. And it's only able to get 4 across at any one point in time. 4, 5, 6. So it's trying to push them across, but it can't get them across fast enough. What's the cell's response going to be? What would you do? If you needed as many of these things as possible, what would you actually do? This is diffusion. That would be perfect, but this is diffusion, so we don't need the energy.
Could they program, like, send more proteins to make them?
Yeah. So they would make more carrier proteins.
Yeah.
Now you guys are going. Oh, it's okay. It's safe. You guys are going to be going into the world of alcohol soon, if you haven't already entered the world of alcohol, because you'd obviously only do that when you're 18. Your body will develop a tolerance to that. Possibly, I don't know, I haven't done the research, but it's possible that part of that tolerance is because your body changes the number of proteins that are affected by alcohol. Either the ones in your brain that alcohol actually slows down, so that it produces more of those, so that it can be more tolerant and keep coordination going, or perhaps it's able to produce more of the proteins that actually break alcohol down. So these responses that the body actually drives are also a key determinant in what's going on. A person who's heavily exposed to a particular drug, whether it's alcohol or cocaine or antidepressant medication, doesn't matter which drug, their body can develop a tolerance to it. I was reading an article just today. There was a group a couple of days ago, a group of 14, 15-year-old boys in Queensland that overdosed on a drug, and they're not entirely sure what drug it is. But the drug that they suspect it could be, it's not actually legal in Australia at all. I can't remember what it's called. But it's very common in Russia. It's used quite a lot in Russia. But one of the problems with it, one of the reasons why it's banned here, is because it not only creates an addictive response, but it also engenders a tolerance. So that people who use it, because it has some medicinal reasons for in anxiety medication, but people who use it develop a tolerance, and so they have to keep increasing their dosage. The problem is that while they're increasing their dosage, their body is possibly, eventually, going to be damaged. So you can't utilize it as a long-term solution because of that tolerance. And it's likely to do with some kind of genetic response. Something similar to what we just talked about. All right, so when we plateau, the body can either, as a medium-term or long-term response, increase the number of protein channels or the carrier proteins. But in the short term, it's just going to plateau. The reaction, the diffusion rate is just going to plateau. Here we've just got a graph, a very small amount of time with this. Simple diffusion follows a linear curve. Simple, is that a question, sorry?
No.
Simple diffusion, what I mean there is it's just diffusion that occurs directly across the membrane. Facilitated diffusion is the one that occurs via carrier proteins or protein channels, and you can see that it tends to plateau. As substrate concentration increases, transport rate eventually flattens off. Now there's a couple of small problems with that graph. Can anyone just identify the biggest problem with that graph, based on what you know about simple diffusion versus facilitated diffusion? Because there is one. Yes?
Does that eventually become simple diffusion? What's it called?
It plateaus?
Yeah.
No. Simple diffusion will always keep increasing. But there is a problem. I'll give you a hint. It's to do with the relative slopes of the graph. What do we know about facilitated diffusion? Is it faster? slower or the same speed as simple diffusion? It's faster. Does that graph show that it's faster? No. They're the same, which is nonsense. That's not true. So although the relationship is true, that simple diffusion is able to continue increasing, whereas facilitated diffusion plateaus, that simple diffusion graph should have a significantly less steep slope to indicate the fact that facilitated diffusion is actually faster. But sometimes people who write textbooks don't think about all of the problems. Right, last slide, then we're on to key concepts, and I want to just go back and look at that activity, and then we'll have our break in between the lessons. So, nature of the substance itself. We've already kind of talked about this a bit. The physical nature and the chemical nature of the substance itself that's being exchanged will also impact on the rate of exchange. So the physical size, who did the nanoparticles, the silver nanoparticles? Do you remember Katie talked about the different shape of the nanoparticles?
Yeah.
So the actual shape of a substance, not just the size, but whether it's a square or whether it's a cube, whether it's a sphere, whether it's an irregular shape, that can also impact the rate of diffusion, even on an incredibly small scale. Small uncharged molecules will typically cross the membrane via diffusion or facilitated diffusion relatively easily. Lipid-soluble molecules are also able to cross the membrane. Now, if you have, let's say you've got a fat, and this is a little bit of chemistry, but you've got your glycerol backbone, and let's say you've got just a, what's that, 1, 2, 3, 4, 5. So you've got a 5-chain fatty acid. So you've got your lipid, and it's all 3 of its side chains are just 5 carbons long. And then you have another lipid, and that lipid is, whatever, 20 fatty acid carbon long, each of them. Which one of those is going to diffuse slower? The 5 carbon or the 20 carbon lipid? Yeah, the 20. The 20 is going to take longer. I think you just misheard.
Yeah, probably. I thought you said which one does it fast.
Yeah, no, which one takes longer? So, yeah, the 20. So it's a combination of physical and chemical. Don't just jump to the conclusion of, oh, okay, well, it's lipid soluble, so it's going to cross the membrane and it's going to do it really, really quickly because it can dissolve in the fatty material around the cell in the phospholipid bilayer. While that's true, larger molecules are still going to take more time. Water-soluble molecules, or at least large water-soluble molecules, and ions require facilitated diffusion. Things like glucose, water-soluble but relatively large, compared to something like carbon dioxide or oxygen. Water itself can move across the membrane, but it's quite slow without aquaporins. Aquaporins are those specialized protein channels for water. Some molecules are complementary to carrier proteins or pumps that move across the membrane that way. So there's a whole range of different mechanisms, and each one is driven by the physical and chemical nature of the substance itself. Particularly, or maybe not particularly, but it's especially important, the physical and chemical nature for protein-mediated or protein-facilitated transport, because the proteins are going to be specific for that, molecule. So if it changes, if something unusual happens to it, there's a potential for something to go a bit wrong. We've got our key concepts.